Welcome to Neurology Grand Rounds, and welcome to our folks who are watching from alternate vistas. So it is my pleasure to introduce Dr. Richard Ransohoff, who is visiting us from currently Biogen. Actually, just until very recently, Dr. Ransohoff was the director of the Neuroinflammation Center at the Cleveland Clinic. And he currently is continues to hold a professorship in molecular medicine at the Cleveland Clinic. And Richard has done a fair amount of his training and career at the Cleveland Clinic, and looks like is now moving on to the East Coast. So aside from being a man in action in pharma, Richard actually has had an incredible career in the field of neuroimmunology. He's his straddling, I guess if you can straddle three different areas: neurology, neuroscience, and immunology. He's led the way in telling us more about what the CNS innate immune cells are really about, what makes them unique, and why knowing how they're unique in their unique environment impacts our understanding of neurological disease. So Richard has won an incredible number of awards and has given lectureships at the ANA. He's won the 2000, let's see, there's so many I had to turn the page over, 2012 John Distel Prize for MS Research. He gave the F.E. Bennett Memorial Lecture to the ANA in 2009. He's a fellow of the American Association for Advancement of Science, and he's also been on the editorial board of a number of journals, including Nature Reviews Immunology, the Journal of Immunology, where he's a section editor, and he is the founding editor of Neurology, Neuroimmunology, and Neuroinflammation. So clearly, Richard has a very important role in this junction between immunology and neurology, and today he will talk to us about a clinically relevant process affecting neuromyelitis optica. So thank you.
Pardon me while I hang myself here. Okay. Thank you for the kind introduction. It's a great pleasure to be here again with friends and tell you about some new stuff we're doing in the understanding of neuromyelitis optica. This started with a series of fundamental questions that we were trying to ask using a blood-brain barrier model. And so first I'm going to talk about the blood-brain barrier model and introduce it briefly. And then everything else I'm going to tell you is new and unpublished and relates to trying to understand the details of pathologic process in neuromyelitis optica, which is really is a brand new disease. It's only 10 years old if you think of when the AQP4 antibodies were discovered. So it's something where we have one big answer and lots of new questions. So I'm going to talk about the blood-brain barrier a little bit and introduce our model, and then mostly go to part 3 and talk about the effects of what's called NMO-IgG, which is the immunoglobulin infraction of people with NMO, and how these IGs function at the blood-brain barrier in our view. It's always worth keeping in mind, I think, this picture when you think about the brain. This is from Zlakovich and colleagues, one of their reviews a few years ago, and you just cannot escape the fact that if you do a cast of the vessels in the brain, it essentially looks like these vessels fill up the entire brain. And in some sense, they do. The guesstimate has been made that no neuron is more than 50 microns from a capillary. So there's almost one capillary for every neuron. Blood-brain barrier needs to be thought of as a multicellular structure, including endothelial cells, 2 basement membranes, pericytes, and astrocytes. We have been trying to understand how leukocytes and endothelial cells interact at the blood-brain barrier for quite some time, maybe about 15 years. And one of the realizations we came upon a little over 5 years ago was that trying to study this process without incorporating flow always gave you the wrong answer. It doesn't give you a limited answer or an incomplete answer, it just gives you the wrong answer. So we decided to try to establish a blood-brain barrier model that incorporated flow. The reason that the answer is wrong if you don't have flow is because leukocytes and endothelial cells interact in order to determine every single trafficking event that any leukocyte does anytime in the body. And those cells are adapted to interact under flow, not under static conditions. So what we do in our model is introduce peripheral blood mononuclear cells, which is monocytes and lymphocytes, with a pump that pumps them across these various membranes here. We have an array of 9 in the current flow chamber. And then this is the efflux here, and this is all done at 37 degrees. So everything is kept at 37 degrees, and the shear stress that we obtain is around 2 dynes per centimeter square, which is right about in the middle of what one sees in postcapillary venules in the CNS. One of the things that I'm going to talk about in explaining how our model works is that we are cognizant of the multicellular nature of the blood-brain barrier. So we have brain microvascular endothelial cells, which you'll sometimes see abbreviated like this. And these come to us from a colleague in Japan, Takashi Kanda and Yamaguchi, who has generated cells from human brain which are immortalized using a temperature-sensitive protein, SV40T antigen. So the cells grow at 33 degrees very nicely, and then if you switch them to 37, which is the physiologic temperature, the T antigen dissolves and absolutely disappears, and the cells differentiate. So it gives you cells that are close to primary cells, but give you reproducible results that are nice to work with. And so we have cells from Takashi Kanda that are both brain microvascular endothelial cells and astrocytes. We also have pericytes, but I'm not going to be able to present those data today. So here's how we co-culture them. These experiments take minimally 6 days to do. So we lay down collagen on a membrane at day 0. We culture the cells, wash and feed, and they expand for about 3 days until they're subconfluent. Then they're switched to 37, so they differentiate. They stay at 37 for about a day and a half. Then they're activated with low concentrations of inflammatory cytokines. These are the endothelial cells. We're mimicking an inflammatory environment. And then on day 6, we do an experiment. So when we have cells from patients, we have to plan 6 days in advance to make sure we're ready to get and work with those cells. So the way this is done is we plate the astrocytes, then we flip the membrane, then we plate the endothelial cells. So the apical aspect of the endothelial cell layer is going to be called luminal. And this is the abluminal aspect of the endothelial cells. They make their own basement membrane. This is the polycarbonate membrane they grow on. These are the astrocytes. Eventually, they fill in. They become confluent. We activate them, and then we do experiments. So this is what the endothelial cells look like with cell tracker green. You don't see anything at the membrane layer except some astrocyte processes, and these are the astrocytes. This is from Yukio Takeshita, who also came over from Yamaguchi with the cells to do a postdoc with me about 5 years ago. The co-culture with astrocytes makes a big difference for the endothelial cells. So I mentioned that we're assaying these cells under flow conditions, which is somewhat stressful, and we also expose them to inflammatory cytokines, which is somewhat stressful. And it turns out that if you include the astrocytes in the culture, the endothelial cells are much, much healthier and much more resilient to this treatment. So when we do these experiments that I'm going to show you right now, we carry them through the entire series of manipulations and fix them and stain them for Claudin-5, which is a component of the blood-brain barrier, and ESAM, which is simply a marker for the plasma membrane in the cells, because the plasma membrane protein. If you handle the cells without astrocytes, this is what they look like simply after the exposure to cytokines. You can see that they vary quite a lot in size and in staining characteristics, and they don't look nice and spindle-shaped as endothelial cells should. This is what they look like in the endothelial cell astrocyte coculture. If you introduce shear stress, then the endothelial cells really look ratty if they don't have astrocytes on board. If they do have astrocytes, they're much happier. So in summary, we have this relatively advanced model with conditionally immortalized cells that are nice to work with. We're co-culturing astrocytes and endothelial cells. Everything is human, which is important because we're interested in human trafficking determinants. We're interested in the molecules that human cells use to get across human endothelium. And it turns out there is a certain amount of variability in these adhesion molecules between humans and other species. So it's valuable to have a human system. We handle the cells under physiological conditions with regard to temperature and shear stress, and the cytokine cocktail that we use is fairly low concentrations. It's 5 units of interferon gamma and 10 units of either TNF or IL-1. It also allows us, this system, to recover and characterize the migrated populations after they cross the endothelial barrier and the astrocyte barrier. So we can ask, what are these cells like once they've entered what would be the CNS perivascular space? The endothelial cells are susceptible to cytokines, and they promote leukocyte migration in response to chemokines. That's an aspect we won't be talking about today. And again, the astrocytes are helpful. So I'm just going to, as an example, give you one snapshot of the type of question that we can use this system to ask. So one thing we can ask is whether leukocytes cross this endothelial monolayer at interendothelial junctions or transcellularly, going right through the endothelial cell. Both types of transmigration have been observed in EM images taken from either people or animals that were undergoing an inflammatory CNS process. But we don't really have any quantitative sense of how often these different routes are used. We know that the leukocytes do not blast through the endothelium and wreck it. We know that there is a highly organized process by which the leukocytes and endothelial cells interact with each other to promote transmigration without destroying the blood-brain barrier and without destroying the endothelial monolayer. But we don't really know very much about the cell biology of the process. So you're familiar with the experimental setup. Here we did a transmigration under flow. The PBMCs were labeled with a dye called calcine, which can be seen here. The blue is DAPI, showing nuclei. And afterwards, we stained with ESAM, which gives us the borders of the endothelial cells. And you can see these nice interendothelial junction borders here. And then, I'm sorry, Topro 3 is for nuclei, not DAPI. So what we saw here was what the lab people persistently call ducks in a row. I guess that's because we're in Boston now. And that means that we found these cells which were stuck on the endothelium after the experiment was over, very reproducibly at the interendothelial junction. And if we quantify this, this is well over 80% of the cells are found at the interendothelial junctions. This is a cell that is on top of an endothelial cell right in the middle, but for the most part, that doesn't occur. And so it's possible to ask, are these guys essentially the losers that didn't make it across and got stuck, and the interendothelial junctions are, kind of a dead end for transmigration, or are they cells that were caught in the act of transmigrating? And in fact, transmigration does occur at the interendothelial junction. And so one way to ask this is to do block face scanning EM and look at the cells that you catch, which are actually in the process of transmigrating. And this is what we found. This is an endothelial cell. This is another endothelial cell. And this is a leukocyte, which is right in the middle of transmigrating. And so this is a picture of how they transmigrate. This is the luminal surface. And you can see this one is sitting on an interendothelial junction. If you flip this over, what you see, so this is a 3D reconstruction of the EM. If you flip it over, what you see is that the leukocyte comes to the endothelial plasma membrane and essentially sort of wraps itself around in order to get across. And so that's what this looks like as you flip it over. The leukocyte essentially fills up every bit of space where it has distracted the two plasma membranes of the endothelial cells from each other. So that's the kind of research that we're using sort of at a basic level to understand leukocyte endothelial interactions. The bulk of the talk is going to use that system, but it's going to address a different question, and that is, what is the nature of the interaction between NMO-IgG, the immunoglobulins in the plasma of people with NMO, and the elements of the blood-brain barrier? So neuromyelitis optica, as you know, it's an autoimmune disease. It really is an autoimmune disease. The disease is caused by antibodies against a self-protein, which is AQP4 water channel on the astrocyte end foot. So it's an autoimmune inflammatory astrocytopathy. You're going to see this cartoon a lot. So the elements are, this is the endothelium, this is the astrocyte, this is the astrocyte end foot containing the AQP4 water channel, and this is NMO-IgG out here on the vascular side in this case. I'm not the artist who did this, and if I were, it would be even worse. So the IgG plasma fraction of NMO patients, which we're calling NMO-IgG, contains this antibody, AQP4 antibody, but they're not synonymous. It's one component of NMO-IgG. For the most part, the antibody is made in the circulation, in the peripheral compartment, by plasmablasts, which are cells that make antibodies. So the questions we're going to ask are, how does NMO-IgG get across to access the water channel on the end foot in the CNS? What are the effects of NMO-IgG on endothelial cells on the one hand and on astrocytes on the other? So again, it's a severe autoimmune inflammatory astrocytopathy. As you know, attacks are often severe and irreversible. Oligoclonal bands are negative. Coexisting autoimmunity is common, 30 to 40%. And many of these characteristics, particularly the negativity for oligoclonal bands and the coexistence of other definable autoimmunities, those stand in contrast to multiple sclerosis. NMO, of course, had been considered for better than 100 years a subtype of MS because they really are distinct among diseases in their episodic nature, and they target same areas of the nervous system, in particular the central nervous system, optic nerves, spinal cord, and in the case of NMO, some of the midline structures as well. The discovery of the IgG biomarker for NMO really revolutionized understanding of this disease. So NMO-IgG is an interesting story. It was discovered as a distinctive perivascular immunoglobulin deposit in the plasma of NMO patients upon incubation with rat cerebellum. So Van de Lennen at Mayo, who runs the serology lab for a variety of autoimmune and paraneoplastic diseases for many years, had been collecting sera and had a sort of a side project in which she incubated these sera with CNS tissues to see if anything happened. She's interested in neurological diseases essentially forever. And so she wanted to see if there were some antibodies out there that would be interesting to discover and work with. After she had gone through about 80,000 samples, she had about 11 positives for this distinctive staining pattern. And so she got her clinical colleagues to get in touch with the people who had sent in these samples, and it turned out that of the 11, 8 of them had NMO. I think one had optic neuritis, one had myelitis, and the other one couldn't be found. So that was pretty impressive, and that really led to the discovery of the aquaporin-4 antibody. The target was reported the year after they reported the clinical biomarker, the NMO-IgG. They found that it was an antibody to aquaporin-4, and now the standard test is a highly sensitive cell-based assay for antibodies to aquaporin-4. And again, it has been rather clearly shown that most of the antibody is produced by plasmablasts in the circulation. So how does this antibody cause disease? It has a plasma membrane surface target, so it is plausible that it could cause damage to cells that have the target. But what does it really do? If you look at the pathology, one thing you can see is that in an NMO lesion, there's no more aquaporin-4. And if you look at the cells that border the lesion, that are called healthy here, what you see is that the aquaporin-4, which usually has a nice linear staining pattern, now looks intracellular and rather vesicular. Another long-standing observation, which hasn't received quite as much attention, is the fact that the vessels don't look normal. They look thick and hyalinized. They don't look like they have a vasculitic process. They are heavily decorated with immunoglobulin, as shown here. To some extent, this would look like a lupus cerebritis autopsy specimen, but these vessels are clearly not normal, and they're abnormal in a way that is distinct from that seen in multiple sclerosis, suggesting something in NMO is acting both towards astrocytes and vessels. This is a scheme that we proposed a while back for NMO pathogenesis at the blood-brain barrier. This is just a sort of speculative scheme to frame hypotheses. B cells in the circulation are making IgGs, including the aquaporin-4 antibody. They get across by an unknown process. They interact in a non-lytic way with aquaporin-4 on the astrocyte plasma membrane. This elicits the production of inflammatory mediators. I'm showing a chemokine here because I like chemokines. To some extent, there is internalization and degradation of aquaporin-4, which, as you know, comes along with the glutamate transporter E2 and also the inward rectifying potassium channel KIR4.1. And ultimately, as a result of this stimulation of the astrocyte to express inflammatory markers, there is additional pathology, including recruitment of leukocytes. The distinctive leukocytes in NMO lesions include eosinophils and neutrophils, quite different and much more destructive than the cells found in multiple sclerosis lesions. Blood-brain barrier breakdown occurs, reactive oxygen species are made, astrocytes are ultimately lost, and in some secondary way, this leads also to loss of myelin. So, IgG is produced in circulation but acts in the CNS. We know from trying to get therapeutic antibodies into the CNS that only about 0.1% of all plasma immunoglobulin gets into the CNS. So how does the antibody get in? And what we decided to ask, because of that decoration of the vessels with immunoglobulin and changes in the vessels, was whether NMO-IgG might contain something that would affect endothelial cells. This could either be a less specific aquaporin 4 antibody, which might see aquaporin 1 on endothelial cells, or it might be a completely separate specificity, based on the idea that a lot of these people have definable antibody-mediated autoimmunity, such as lupus or Sjogren's syndrome. And then secondarily, we wanted to ask, once NMO-IgG and aquaporin 4 antibodies get into the CNS, how does their interaction with astrocytes affect the blood-brain barrier? So you've already seen all this stuff about the coculturing. I won't go over it too much. You're going to see two colors of astrocytes, because in addition to astrocytes that we use in our routine studies, we have a separate culture line of astrocytes which are transfected to express aquaporin 4. Human astrocytes in culture express aquaporin 4 very poorly. And so if we want to study the role of aquaporin 4 antibodies, we need a cell line that has aquaporin 4. So the purple diamonds have aquaporin 4. The little blue things have are the endothelial cells. And while we're talking about colors, I want you all to notice I have Seahawk colors on today, even though I'm coming from Boston. For those of you that don't know, the Seahawks are a football team that plays here. So in these experiments, we put NMO-IgG on the vascular side. That means in contact with the endothelial cells. Or we put it in contact with the astrocytes. And then we ask about effects. When I talk about NMO-IgG in these first experiments, I'm talking about material that was prepared by Van der Lennen. It's a pooled sample from 50 plasma exchange samples. And we have control IgG from a healthy human serum pool. They were made at the same time in the same way in the Lennen lab, and they're provided to us initially coated at the same concentration. So they're nicely comparable. And they use protein G, so they're pulling out only IgG. This is just a quick look at what the cells look like. The only thing to notice here is the endothelial cells are pretty flat. The astrocytes are not. And the astrocytes extend processes towards the endothelial cells that, if you wanted to be very optimistic, you might say these look like astrocyte end feet in vitro. So the first thing that we assayed was something that endothelial cells do readily when they see an inflammatory stimulus, and that is they upregulate an adhesion molecule called ICAM1, which helps them interact with leukocytes. So if you don't stimulate the endothelial cells and stain for ICAM1, there's nothing. If you stimulate with the cytokines and then stain, there's quite a lot. And so here's the experimental setup. This is the endothelial cells and the aquaporin 4 astrocytes, which we call A4. And here's two duplicate cultures of those cells. These astrocytes do not have aquaporin 4. And what that's going to do is help us control for the possibility that when we put the NMO-IgG on the top in contact with the endothelial cells, if a little bit leaks through and stimulates the astrocytes, as we know it does, then that might feed back and turn on the endothelial cells. So this is a negative control. This is a negative control just because it's getting the control IgG and so forth. And here's what we saw. If you put the NMO-IgG in contact with the endothelial cells as compared with the control, regardless whether there's any aquaporin-4 on the astrocytes, they turn on ICAM1. Those cells are responding to something in NMO-IgG, and it has nothing to do with the presence of aquaporin-4 antibodies. So we found that very exciting. So we then wanted to sort of go for a more fundamental type of inflammatory response, and that's nuclear translocation of NF-kappa B. NF-kappa B is a transcription factor involved in almost every kind of inflammation, and the way it acts is it sits in the cytoplasm. When the cell is stimulated, it goes to the nucleus and turns on a wide array of inflammatory genes. So here you see NF-kappa B in the cytoplasm of these endothelial cells. If you hit them with TNF, it goes right into the nucleus, and you can see the nuclear staining here. So similar experimental setup, but without astrocytes. And again, what you see here, this is actually from the literature. These are our cells, and you see these very pretty sort of empty nuclei here with cytoplasmic staining. If you come with TNF, all of that NF-kappa B goes right into the nucleus. If you expose these cells to control IgG, nothing happens. If you expose them to NMO-IgG, it goes right into the nucleus. So these cells are being strongly stimulated in an inflammatory way by something in NMO-IgG. That would imply that there's some target on the endothelial cells. So we simply stained the cells without fixing them. We did live cell staining so that we could see surface structures. When we use NMO-IgG, it stains the endothelial cell. You can see it's a cytosolic or cytoplasmic target, not in the nucleus. Control IgG does not interact with the endothelial cells. Aquaporin-4 antibody does not interact with the endothelial cells, so it isn't a cross reaction with another aquaporin. And the next question is, if this stuff is present in bulk IgG from 50 NMO cases that were sick and underwent plasma exchange, what about individual cases? So Dr. Lennon's lab produced a whole series of samples for us that either came from individual NMO people who got plasma exchange, or people with lupus, or controls who didn't have either one, and where she just isolated the Ig fraction. So do IGs from individual cases show these effects in staining patterns? What you would predict from NMO-IgG, if we're correct, if what we're seeing has any meaning for the individual patient, is that stuff from people with NMO might bind to endothelium. It should also bind to astrocytes by an aquaporin-4 dependent means. Samples from people with lupus might bind to endothelium, because we know people with lupus have endothelial active antibodies, such as phospholipid antibodies, or what used to be known as lupus anticoagulant. And the control should bind to nothing. Samples from people with lupus should not bind to astrocytes, regardless whether they have aquaporin-4. So here are these are the controls. This is just the phase contrast of the cells. So controls don't bind to endothelial cells. NMO-IgG bulk does, and these two individual cases do. These lupus cases do. These are two controls, they don't. And by the way, this is all done blinded. So Vanda simply sent us a bunch of tubes, we did the staining, and then she sent us the code. Again, here's one from NMO, one from lupus. This is an NMO that doesn't have that reactivity. Here's another lupus one that does. So this endothelial reactive staining pattern was characteristic of people with these inflammatory diseases in a very common fashion. This sort of astonished us. But we find it exciting because it suggests that there's a two-hit mechanism for NMO in which one antibody binds to endothelium and activates the endothelium, allowing transit of the aquaporin-4 antibody. Then the aquaporin-4 antibody binds to the astrocyte and initiates a cascade which results in lesions. What about the aquaporin-4 staining? So these are astrocytes now down here. This is the incubation with the control IgG. It turns out when we incubate with pooled NMO-IgG, we see somewhat that vesicular pattern of staining that you saw in the tissue. This drives internalization of aquaporin-4. This NMO-IgG does the same. This one simply stains the plasma membrane of the protein, of the cell. Here's a lupus sample that interacts with astrocytes but should not. So that's number 7. Keep that in mind. Okay? And these are all aquaporin-4 positive astrocytes. And these are the rest of them. These lupus samples don't, but remember these did stain endothelium. These NMO samples do. This one stains the cells. This one drives internalization. So they're not all identical. What about the negative controls, the aquaporin-4 negative astrocytes? So nothing stains them, but look at number 7. Number 7, the lupus case, has something that stains astrocytes, but it doesn't stain aquaporin-4 specifically, it just interacts with astrocytes. So that's a non-specific staining. This is a summary, and basically it's what I told you. The pooled NMO-IgG binds endothelium, induces ICAM, didn't have time to show you that, binds to aquaporin-4 astrocytes, does not bind to the control astrocytes. The control IgG binds nothing. The individual NMO-IgG cases look like the pools, and the lupus cases, for the most part, have the vascular reactivity, which activates the vessel, but does not bind astrocytes, either control or aquaporin-4. So there may be a very broadly distributed antibody in the plasma of people with inflammatory disease that binds and activates endothelium. And this is going to read out as a brain disease if there is another antibody in the plasma that can cause brain disease. So this may be true in these non-infectious encephalitides, the NMDA receptor diseases. Obviously NMO is one that we consider, maybe some paraneoplastic diseases and so forth. So how does NMO-IgG affect endothelial cells? It binds them and it activates them. One thing I didn't show you is that using labeled IgG, we can show that this binding and activation is associated with increased transit of immunoglobulin from the luminal side to the abluminal side. So letting in the aquaporin-4 antibody is certainly a possibility here. Now, does this have anything to do with leukocyte transmigration? That's essentially where we started. And to ask that question, we put the NMO-IgG in contact with the astrocytes and ask about the effect feeding back towards the endothelial cells. Remember that astrocytes are responsible for maintenance of a healthy blood-brain barrier in adults. So what happens when you perturb the astrocytes? So here we have the same set of cocultures, but now the NMO or control IgG is going in contact with astrocytes. And so we're looking at the dependency of effects on the presence of aquaporin-4 on astrocytes. And what we see is that when you put NMO-IgG in contact with astrocytes that do have aquaporin-4, something happens to the endothelium which approximately doubles leukocyte transmigration in a 1-hour assay. So it's a very potent effect. I can tell you this is quite a potent effect in our system. It's the equivalent of stimulating the cells with inflammatory cytokines. So something is happening with the astrocytes that activates the endothelium and promotes leukocyte transmigration. So what is that? I told you that we like chemokines. These are some chemokines. Those are molecules that promote leukocyte transmigration under many circumstances. And we had an array of them in which we were interested. And so we measured them. And we measured them specifically by scraping away the endothelial cells and asking about upregulation of all of these different chemokines by looking at the messenger RNA by quantitative PCR. 2 of them were greatly upregulated. The rest were not affected at all. This is CXCL8, which is also known as interleukin 8. It's a neutrophil-specific chemokine. This is CCL2, also known as MCP1, monocyte chemoattractant protein 1. It's a monocyte chemoattractant. It can also, under some circumstances, attract eosinophils. So it seemed plausible to us that this might be an effect which could relate to the inflammatory infiltrate in NMO lesions. So the second summary, looking at the effects of NMO-IgG at the blood-brain barrier, is that when the antibody gets across and interacts with the astrocyte makes chemokines, which are typically taken up by the endothelial cell and immobilized on the luminal surface where they interact with cells in the bloodstream and promote their transmigration right at that spot. So how does this happen? What is the astrocyte doing that talks to the endothelial cell? So in order to ask that question, we took specifically, I'm so happy to say that's not me. We took the astrocytes that had aquaporin-4, exposed them either to NMO-IgG or control IgG, and then we asked about an entire array of chemokines, cytokines, and growth factors, particularly growth factors that might act towards endothelium. And these are basically chosen from things that we thought might mediate some of the effects we were seeing. Only one of them was upregulated, and that's interleukin 6. Interleukin 6 is known to be active towards endothelium. Mice, in fact, that have chronic overexpression of interleukin 6 in the CNS have a disrupted blood-brain barrier from early in life. So this was an interesting cytokine to consider. And it was interesting to us that there was a great deal of specificity in this response. There's 23 different factors here, all of which can be made by endothelium, but in this case, only interleukin 6 read out. This is just to show that the interleukin 6 protein, not just the message, was induced. And it also shows you that this is not simply due to some contaminant like LPS in our NMO-IgG, because when you incubate the non-aquaporin-4 astrocytes, remember we only had aquaporin-4 astrocytes here. If you assay the non-aquaporin-4 astrocytes, nothing happens. So this is not a non-specific contaminant. This requires aquaporin-4 on the astrocyte. Just a little bit of digression about how IL-6 works. For the most part, IL-6 signals to cells that have the IL-6 receptor, which makes sense. And that's a limited number of cells, some inflammatory cells, hepatocytes. IL-6 drives the acute phase response by hepatocytes. So this is IL-6. It actually is shaped like a barrel. This is the IL-6 binding receptor. This is what generates the signal, GP130, which is on every cell and generates many different signals. But all by itself, it can't generate a signal. It needs a stimulus of some factor binding to some binding protein in order to elicit this signal. So this is called classic signaling because the IL-6 receptor is on the cell. There is also a soluble released form of the receptor that is found in blood and in body fluids called soluble IL-6 receptor. In the presence of high concentrations of IL-6, this thing can bind to the soluble receptor, interact with GP130, and elicit a signal. And that's called trans-signaling. We know that endothelial cells don't have the plasma membrane receptor for IL-6, so we had to look at a soluble IL-6 receptor trans-signaling system. So we put endothelial cells together with IL-6 and soluble receptor for 2 hours, and we looked at the chemokines that they made after astrocytes saw the NMO-IgG and looked at that by PCR. And we also looked at permeability by looking at transit of labeled dextran. And indeed, in the presence of soluble receptor plus IL-6, the endothelial cells made CCL2 and CXCL8, same guys that they made when we stimulated the astrocytes with NMO-IgG. And furthermore, their permeability was increased. Now, does astrocyte IL-6 have anything to do with this enhanced leukocyte transmigration? To ask that, we took endothelial cell astrocyte cocultures, gave them control or active reagent, the NMO-IgG, and then in the next well, we added the antibody to the IL-6 receptor. And indeed, when we gave them NMO-IgG, leukocyte transmigration went up, but if we blocked only IL-6, it went down to the baseline level. So IL-6 is necessary. And it also turns out IL-6 is sufficient. If you simply expose these cocultures to IL-6 before transmigration assays, again, you get a significant increase in transmigration. So that's the whole story. NMO-IgG selectively induces IL-6 in aquaporin-4 containing astrocytes by aquaporin-4, and IL-6 feeds back to the endothelium, activates the endothelium, and promotes leukocyte transmigration. And some of you may know that the IL-6 antibody, tocilizumab, which is a clinical stage product, has been tried in very limited clinical trials for NMO and seems to have a potent therapeutic effect. And there are some much larger clinical trials now ongoing, primarily in Japan. So IL-6 trans-signaling increases. How does the leukocyte transmigration work? The endothelial cells make chemokines which promote it, and the chemokines enhance leukocyte transmigration. Also, IL-6 trans-signaling directly decreases barrier function. So putting it all together, NMO-IgG at the vascular side upregulates ICAM in part via NF-kappa B. There may be other signaling, and activates the endothelial cells. This will then promote the transit of aquaporin-4 antibodies to engage these aquaporin-4 water channels on astrocyte end feet. That's going to elicit IL-6. IL-6 feeds back and further activates the endothelium, now to promote leukocyte transmigration. And that cellular lesion is the definitive lesion of NMO, which is a highly destructive process. So an IL-6 receptor antagonist inhibits leukocyte transmigration and also shows some evidence of therapeutic efficacy in limited clinical trials, and is not a Biogen product, I might say. So NMO-IgG contains IGs which directly modulate endothelial cell function and are not targeting aquaporin-4. And this is our group. Just by coincidence, the main people who worked out the blood-brain barrier model were Bunny, Birgit and Brian. So they're known as the BBBs. But they couldn't have done all this without Alyssa, Hayan, and mainly Yukio, who did all of the NMO work that I showed you. So thank you very much for your attention.
Toggle the mouse pad, there may be questions behind you on the screen.
Oh. Okay, well, so you want me to do this?
Yeah, and then minimize
Do that. Like that?
it. Yeah,
Okay.
good. Okay, just go.
Okay, to the rescue. Oh, I have to look at myself. That's embarrassing.
We can look at Dr.
Yeah, hi.
I have two distinct questions for you, if I may. So one is, why are the plasmablasts making these antibodies? Is that what is initiating the disease?
Okay, and the other?
The other question is, why is this disease picking out the optic nerves and the spinal cord?
Okay. So it says right here, please repeat the questions. So I'm going to do that. First question was, why are people making an antibody to a self-protein? And that's way above my pay grade. I don't think anybody knows. I would mention that these are people who are predisposed in many cases to make autoantibodies. So there are decent sized case series, for example, of people who have NMO and also have myasthenia gravis. So they're making acetylcholine receptor antibodies as well. I did mention also that a decent number of these people have lupus or have Sjogren's syndrome. So there's a fundamental problem here with control of B-cell reactivity. AQP4 is not only found in the CNS. In the CNS, it's really restricted to astrocytes, but it is also found in kidney in large amounts and also in skeletal muscle. So in addition to asking why optic nerves and spinal cord, it's also worth asking why not kidneys and muscle. You almost never see any kidney pathology, as best I know. Muscle pathology that's clinically articulate is also not seen. There are occasional retrospective findings of what they call hyper-CKemia, where the muscle creatine kinase on a random lab is found to be very elevated, and then it goes away. And it's not associated with evidence of myositis and so forth. So it is not clear why people make these antibodies. Second question was why spinal cord and optic nerves. The presumption is that that's because that's where there's a lot of AQP4. So the areas that are seen to be affected in pathology are areas where there's a lot of AQP4. You find a tremendous amount of non-lytic astrocyte pathology over the convexities, for example, because there's a lot of AQP4 in the glial limitan superficialis astrocyte processes. There is occasional pathology in area postrema, which causes intractable nausea and vomiting. So it seems to be that's where there's a lot of target. But there may be other factors as well. Yeah.
Yeah, given that there have been some notable cases of myasthenia gravis and NMO coexisting, do you think there's any role for the thymus gland?
Yeah, what a great question. So the question was, given that there's coexistence of myasthenia and NMO, is there a role for the thymus? The only way that anybody would, I think, be able to ask that is by doing thymectomy. And I don't think that has been undertaken yet, although there may be activities along those lines that I'm not aware of. There is certainly always a cooperation between T cells and B cells to elicit antibody production. And there are clearly T cell epitopes for AQP4 that can be defined in human beings. So there may be TB cooperation and there may be a thymus story there. I'm not aware of anything that would link NMO to, for example, thymoma or even thymic hyperplasia, however. Yeah.
Just from an engineering perspective, just for a naive question, when you, with your blood-brain barrier model, are there any other components that in a fantasy world you'd like to add to that? Is there?
Right.
You wonder about?
Right. So, is there anything else I would like to add? We are in the process of setting up at least co-cultures, including pericytes. And that's because when Yukio went back to Japan, he was succeeded by Fumitake Shimizu, who did the staining of the individual cases. And he brought with him pericytes, which he had developed when he was in Japan. So we're certainly going to be doing endothelial pericyte co-cultures. And ultimately, I don't see a reason why we shouldn't do all three. The other project that's ongoing along these lines is to go from a macrofluidic device, which this is, and which really doesn't support high-resolution multiphoton imaging of the interactions, to a microfluidic device, which will. So I showed you the one 3D EM. We have several of those. They're very informative. The 3D reconstructions are pretty, but ultimately we want to watch these cells interact. One thing that leukocytes do during transmigration, I think most of us are aware of the idea that they undergo rolling and then activation and then arrest and then transmigration. In our limited imaging and also very heavily in the literature, it turns out that after arrest, they crawl around like crazy, these leukocytes, on the endothelial luminal surface. And it is fascinating to think about what they're looking for. What are the signals that motivate this crawling behavior? They crawl as often against flow or perpendicular to flow as with flow. So there's an entire other story there to be uncovered. So that's my fantasy. Hi, Gwen.
Hi, so taken to defining the clinical syndrome of neuromyelitis optica based on the length of the spinal cord lesion, and some of those patients are AQP4 antibody negative.
Yes.
Do you think that this mechanism is present in those patients, but just some other antibody that we haven't identified yet, or do you think that is just a different disease?
Right. So the question is, what about seronegative NMO? Is it a different disease, or what was the other possibility?
So it's some other antibody that would
Oh,
cause astrocytes to
yeah.
produce IL-6.
Right. Is it the same mechanism or is it a different disease? So the state of the art right now, and it really is very time limited because it's a pretty dynamic field, which is evolving before our very eyes. State of the art right now is that you have to take into account that in the case of working up a patient with suspected NMO, often based on longitudinally extensive myelitis, once negative does not mean always negative. It is Very important to realize that titers of these antibodies clearly fluctuate so that you need a certain number of tests, I don't know what the number is, to say that they're really negative. Second, the nature of the test matters. So this cell-based assay using recombinant AQP4 expressed by a cell, incubated with serum, and then often analyzed by flow cytometry or by immunocytometry. That's the gold standard now. That's the most sensitive and specific test. So tested an adequate number of times, about 85% of people with the syndrome are going to be AQP4 antibody positive. So we'll put them off to one side and say the rest of the people are truly NMO that's AQP4 antibody seronegative. A certain number of those people turn out to have MOG antibodies, and the MOG antibody story in terms of pathogenesis, really even the role of pathology, is unknown, but there are enough labs that have turned up these MOG antibodies that there is clearly a population of people that has MOG antibodies, and the current evidence is that the behavior of their disease is quite different. They are much more likely to have monophasic disease. The clinical implications of AQP4 antibody is that the person needs to be treated and that they will recur if not treated. The MOG antibody is the flip. It's less likely that they will recur. It is less likely that the attacks will cause non-recovering symptoms. They will do better over time. So there are at least 2 antibody specificities associated with somewhat distinguishable syndromes. But until somebody comes up with another antibody that we can use as a tool to investigate routes towards pathogenesis, I think we're only speculating. You remember how long myasthenia was only associated with acetylcholine receptor antibodies for decades before Angela Vincent came forward with MuSK antibodies. So I think we just have to wait and see what emerges. I should mention everybody that was studied in the data that I showed you was AQP4 antibody positive. Yes?
Have you observed that the entry of NMO-IgG is ICAM1 dependent?
Oh, no. So the question is NMO-IgG entry into the across the barrier dependent on ICAM? Have not asked the question. There's certainly an association. The activating event, which upregulates ICAM, is also associated with increased transit of labeled Ig across our endothelial barrier. But since we don't know anything about the signaling except that it's associated with nuclear translocation of NF-kappa B, we really don't know what the mechanistic links are among the things that we're assaying.
Is natalizumab useful at all?
Yeah, good question. So is natalizumab useful at all? Not only is it not demonstrably useful, but there are cases, including one in my practice, who did worse. So I think it's a very provocative feature of these 2 diseases that putting natalizumab into the system almost has divergent effects. So what does natalizumab do? It keeps cells from exiting the vasculature. And in fact, in people receiving natalizumab, you find increased numbers of lymphocytes, right? And if you assay the lymphocytes, you can find that there is a somewhat higher degree of, for want of a better word, activation of those cells that are retained in the periphery, right? So I think it makes clear that the MS therapeutic effect is mediated by keeping cells out of the CNS. I think it makes equally clear that the lack of therapeutic effect in NMO cases is likely to be because these cells are trapped in the periphery, and that may generate more NMO-IgG. So it says NMO is a disease in which the pathogenic principle is generated in the periphery and enters the CNS as an antibody, not in a cellular form in a way. Yeah.
Given all these antibody-antigen interactions, do you think there's a role for complement here? And is the NMO antibody always IgG or is it ever IgM?
So is there a role for complement, and is it always IgG or is it ever IgM? Role for complement is for sure. The sort of final common or final terminal event for most of the astrocyte pathology is complement fixation and death of the astrocyte. The sublytic pathology is antibody binding and perturbing plasma membrane signaling by cross-linking molecules on the surface. But once complement fixation occurs, then true astrocyte loss ensues. And there's a tremendous amount of, not only is there a lot of complement in the lesions, but there is quite a successful, I think, phase 2 trial that was carried out by eculizumab, which is a C5 antibody. So getting rid of complement has pronounced therapeutic effects. The concern with therapeutic development along those lines is that eculizumab has been released for the treatment of paroxysmal nocturnal hemoglobinuria, right, which is one of these complement defense protein deficiency. And as is the case when somebody releases a drug for a niche disease, it's like $100,000 a year or a week, I don't know, but it's very expensive. And so developing it fully for treatment of NMO is going to be challenging.
We're actually doing the eculizumab myasthenia gravis study.
Wow.
Just started producing.
Wow. Yeah. Actually, myasthenia is the best sort of scientific comparator for NMO in my view, because if you go back 30, 40 years in the myasthenia literature, and people were beginning to think, what does the acetylcholine receptor antibody actually do? Does it modulate the receptor off the surface? Does it block the receptor? Does it fix complement and destroy the membrane? The answer turns out to be yes to all those. And that's the thinking that informed the people who were studying pathogenesis of NMO. Thanks a lot.
Thank you so much.